So this is what happens when the sodium blue disyl sulfate is placed in the protein, and it partially denatures the protein itself. Okay, so there is a migration based on the charge ratio. So the smaller the size of the protein, the faster it will migrate towards the cathode. Okay. And there is also a visualization which uses CBB, silver, or fluorescent dyes. So there's also a quantification or determination of purity on the purified sample of the protein. So we can also get the protein size, the estimate protein size of the protein in the electrophoresis, um, in the gel electrophoresis, I mean. So another one is that we can detect or we can release know the protein when we use isoelectric focusing. So it is basically based on the charge, specifically based on the isoelectric point of the protein. So the pH value at which protein carries no net charge. So in this isoelectric focusing, there is a use of the IEF matrix. It is a gel strip containing an immobilized pH gradient. Charged proteins migrate to either cathode and anode, crossing different pH steps. When migrate to their isoelectric point, protein will carry no net charge, therefore, and there will be no more mobility in an IEF device. So, there is an immobilized pH gradient gels. That's the matrix. So that is the IPGs are the acrylamide gel matrix, co-polymerized with a pH gradient. Okay. So as you can see, so when the protein sample is injected or placed here, so there is a movement or migration. So when the isoelectric point of the protein will be achieved in that particular pH level, it will not move further. So we can now determine the isoelectric point of that protein. So the protein has specific isoelectric point. So the pH levels is from the pH 9 decreasing to pH 3, from the basic to acidic pH levels. So there is another visualization method, which is the immunoblotting, or also known as the Western blotting. Okay. Now, the immunoblotting, it is a rapid and sensitive assay for the detection of and characterization of proteins that works by exploiting the specificity inherent in antigen antibody recognition. Basically, it uses an antigen antibody recognition. So the antigen the protein serve as the antigen, while the antibody is the one that will bind to the antigen. It involves the solubilization and electrophoretic separation of proteins, the glycoproteins or lipopolysaccharides by gel electrophoresis. Meaning there is, before immunoblotting, there should be an electrophoresis that should be conducted. So let us know why there is a electrophoresis. It is really important. So the immunoblotting technique has been useful in identifying specific antigens recognized by polyclonal or monoclonal antibodies and is highly sensitive. This unit provides protocols for protein separation, blotting proteins onto membranes, immunoprobing, and visualization using chromogenic or chemiluminescent substrates. So this is now the process of immunoblotting for specific detection. So as you can see, that you should conduct electrophoresis so that you can really separate your protein and discard, so that you will know, you can assure that there is a separation of the charge. There is now a separation or the migration of your proteins. So it is electrophoresis. This is the polyacrylamide gel, and it will be blotted onto the membrane. The membrane that are used specifically sometimes are nitrocellulose that will really bind the protein bands in the membrane. So this membrane now will be subjected to antibody detection, and this will be incubated with the antibody. So this antibody will bind to the proteins that we are targeting of, or targeted proteins that you want to detect specific protein. Okay. After incubation, sometimes there will be washing of this solution containing the antibody. Okay. So third is there will be an incubation of the enzyme-linked antibody. So this enzyme-linked antibody will now produce color in order to um, project or detect. You can now see it in the color formation. So, which of these proteins are really your targeted proteins in the polyacrylamide gel. Now, if you now know um, your targeted proteins, you can now excise your targeted proteins in the original SDS polyacrylamide gel, and you can further analyze it. So that's the specific detection of your protein. You should know the right antibody in order to detect your antigen, which is your targeted proteins. Okay, so you can also use radioisotopes to label and detect proteins. Okay. The next one is autoradiography. It is an imaging technique. So you can get an image about the structure of your proteins. You not know that Franklin was the one who conducted the autoradiography on the structure of the DNA. That is why when Francis Crick and James Watson um, know or um, have looked the result of Franklin, so they have interpreted it and they really deduced that it is really double helix in structure. But it was really Rosalind Franklin who conducted it for the imaging technique of the DNA. So in the in vitro process, it involves the isolation of cellular components such as DNA, RNA, proteins, or lipids, followed by labeling with suitable radioisotopes. Now, in vivo, I say in vivo, in the living for example, it can be in humans or it can be in the laboratory mice I mean. So in the in vitro, it is in the glass. You can perform an experiment in the test tube. But in vivo, you can perform it in the living system or in organism. So the radioisotopes are coupled with radioactive tracers and administered orally or via injection. So those radioisotopes will then bind and be distributed within the body, and then it will then be evaluated in thin tissue or whole body cryosections. Okay. The principle of this radiography imaging technique is the precipitation of silver atoms resulting from the ionization of a silver halide. So by their radio labeled samples. And this silver halide or the AGX are light sensitive compounds commonly used in photography. That is why you can really have that image or the structure of your proteins. They are generally suspended in the gelatin photographic emulsion. And this silver halide is individually encapsulated in the gelatin and functions as an independent detector of radioactive decay from the radio-labeled sample. So it does not need another reaction in order um, to detect its color or the light itself, but mismo ang silver halide because it is sensitive to light, it can really have that photographic image. So when we want to analyze the proteins, once radioactive particles hit the gelatin emulsion, this silver halide is reduced, resulting in the production of insoluble silver crystals. Okay. How about the spectrometry? So what data can we get in this spectrometer? This is an analytical tool useful for measuring the mass to charge ratio. Basically, you will know the exact molecular weight of your unknown protein. So if you have a newly discovered protein from the microorganism and you wanted to know its properties, mass spectrometry is a powerful analytical tool in order to deduce or to investigate the molecular weight of the sample. So, mass spectrometers can be used to identify unknown compounds via molecular weight determination, to quantify known compounds, and to determine structure and chemical properties of molecules. Okay. We have three different parts of the mass spectrometer. First is the ionization source, which the molecules are converted to gas phase ions. So it is it is done using external electric and magnetic fields. Then second is the mass analyzer. Now once ionized, the ions are sorted, separated according to mass to charge ratio. So after the ionization, there will be an analyzing of the mass in order to there is a separation according to mass to charge ratio. Then there is now a detection system. The separated ions are then measured and sent to a data system where the mass to charge ratio are stored together along with the relative abundance. The mass spectrum is simply the mass to charge ratio of the ions. So plotted against their intensities. So each peak in a mass spectrum shows a component of unique mass to charge ratio in the sample. So this is now the illustration of the mass spectrometry. So the sample will be injected here, and then there is conversion from the molecules to ionization to gas phase ions. Uses the electron beam source and the heater that vaporizes the samples. The ions are accelerated and detected. You see here in this one. So magnetic field deflects lightest ions most. Okay, so this is now the graph that is reflected in the computer. So the atomic mass of your proteins, okay, of the ions itself, so that you will know the molecular weight of your sample. So the last one is the proteomics. Now, what is the simplified definition of proteomics? It is the systematic studies of the protein structural and functional changes. Now, when I say systematic studies, it is not just a one-time event of study of the protein, but the changes of the protein it undergoes under different conditions based on its structure and its changes in function. So those things that I have discussed, the electrophoresis, the immunoblocking, the isoelectric focusing, and the autoradiography, the mass spectrometry, those are tools that are used in the proteomics. The two-dimensional electrophoresis is a tool for protein expression comparison. So between two systems, and the spectrometry or spectrometer is a method that uses an instrument called mass spectrometer to determine the precise mass and that sequence of the proteins. So the proteomics itself is the different studies of the proteins, the changes based on the structure and the functional. So, for example, if you want to know the changes of the protein structure and functional changes in a plant under different conditions, that will be called all the proteins that are involved in the ripening, all the proteins that are involved in the changes of color of your the leaf. So the biochemical system in your plant should be investigated. So that is why you have to really use these things, use this powerful tool in order to detect the changes of your proteins and its functions. That's the overall study of the proteomics. So this will be the last slide of this topic, the protein biochemistry, the analytical methods, and its visualization. So next week, I will now give a quiz for the lesson one, which is the introduction, and then the biosecurity laboratory design in this protein biochemistry and the analytical methods. Then also on Monday, we'll have a quiz for the lesson one and two. So your quiz can be accessed in the Moodle. Okay, so as we have was what we have agreed that you have 24 hours to answer your quiz. It will be a long quiz because it composed of the lesson one and two of the module one. So thank you so much class for watching this video, and I hope that you will really watch it and you will really understand the principle of the analytical methods on the separation and detection of the proteins. If you have any further questions that or not yet you have not understood or you want to clarify something, don't hesitate to message me, or you can raise it on Monday on our discussion part. Okay, so thank you so much, class. Thank you for watching this video, and I hope you will learn something based on the analytical methods of the proteins. Thank you.